question?
The environment.
Is the environment also epigenetic? For example, diet and UV exposure, is that also epigenetic?
Yeah. So yeah, I think you'll hold the question. So those type of factors like the environment, temperature, pH, and your diet and what you might have been exposed to in terms of radiation. So yes, those will alter the epigenetics of these cells. Now, microRNAs are one of three aspects of epigenetics, but they're very important for, and they're very stable effects on epigenetics. So in other words, exposure to sunlight might affect the other epigenetic factors that are involved. And I don't want to go into all those. But yes, those UV light can actually damage your DNA and change the sequence and cause a mutation. So it's not fair to say that, for example, a sunburn is only an epigenetic effect. You hope it's only an epigenetic effect. If it causes a mutation, you may end up with melanoma. So yes, you hope it's something. So this is just an example of how a brilliant group of scientists who published a paper last year took what we knew from zebrafish and their microRNAs and their epigenetic mechanisms and converted that to potentially a new therapy to regenerate the heart. Now, fish can regenerate their fins, and if you amputate 20% of their heart, they can regrow that heart. Mammals, including mice, cannot. Mice, humans, rats, none of us can do that. And the reason is that our heart cells don't divide very well. So we're kind of born with our cardiomyocytes, and once we've grown, at least past adolescence, we have very little to no capacity to replace those. There's been a debate for many, many years about cardiac stem cells. But generally, the bulwarking muscle cells of your heart do not divide. So if they don't divide, you can't regenerate. To regenerate, you need to have cell division. So anyways, the zebrafish can. Their cells can. So they looked and said, why can the zebrafish do that and a mouse cannot? And they found these microRNAs that were controlling the process. Bottom line is you've got to get rid of those microRNAs or your heart won't regenerate and your cardiomyocytes won't divide. So they said, okay, well, if I can see that happen in a zebrafish, what if I did that, experimentally, I knocked down, I got rid of those microRNAs that control cardiac regeneration in the mouse. And they did. They actually caused an infarction in the mouse's heart, and the controls was left with scar tissue. And in the mice where they knocked down these microRNAs, they got complete regeneration of the heart. To me, that's exactly what we're here to talk about. Translating information from the animal kingdom and turning that into a molecular mechanism that's highly conserved, that's safe, that can be used to treat mammal in vivo, not just in the culture dish. Next slide. So the question is, still remains at this point, how are you going to translate all this information to humans? Because you're not going to translate that information from a mouse to a human, because mice control their cells using different microRNAs than humans. They're not the same. So now we need to find a way of testing our ideas safely in patients that are more like us. And that's where the veterinarian perspective comes in. 99+% of all research is done in animals, is done in laboratory animals that are genetically modified in some way to get the appearance and the phenotype that we want, the biology that we want, or we actually end up, scientists injure the animal experimentally, give them tobacco smoke or whatever it is to create the injury model. And then we have this gem over here, all these naturally occurring disease models in pets that are just begging to be studied and treated with, new approaches. There's a lot of dogs and a lot of cats. 78 million, 80 million, horses, birds, also wildlife. A lot of veterinarians, teaching hospitals, and at Tufts alone we see over 40,000 cases a year. A lot of those cases come to us because they're hard to treat, they're during emergency hours, or they're refractory to standard care. Next slide. So this is probably the most important slide, and I want to finish in 5 minutes, so let's see if I can do that. The take-home message of why pets are so interesting as animal models, disease models, is that they have similarities to humans that rodents don't have, that are really critical to predicting the outcome of how a new therapy or a new lab test is going to perform in humans. And there are many, many new advances that are occurring in this field all the time. And if you don't have an avenue towards advancing those therapies, they're going to get stuck and bottlenecked through the laboratory. So what we're proposing is that some of the similarities in the dogs versus humans that are lacking in the rodent are important. For example, the rodent is actually phylogenetically, meaning in terms of evolution, slightly closer to the human than the dog. And I'll show you a slide of that in a moment, which is surprising, right? We don't look like a rat, but our ancestors look like a rat. However, our pets live in exactly the same environment. They have a closer body size and anatomy and physiology. Lifespan is compressed compared to the human, so that gives the advantage of studying something over a short period of time, but it's still fairly long. Their gene homology is much greater than what you see if you compare rodents and humans. Genetic diversity is much greater. And just look at the whole breadth of dog breeds. It's stunning. There's no other animal out there that has morphologically such variety, except maybe fish. But in terms of mammals, I don't think there's anything else. They respond to toxins very similar to humans, but from our perspective, this is the winning aspect of why we should be studying pets and why we are studying pets, in particular dogs, is that they get naturally occurring diseases that are very similar in terms of the symptoms, the pathology, like what you see under the microscope in their tissues, and in terms of their time course and survivorship aspects, so very similar to humans. The way that chemical and protein biomarkers appear in the blood and other biofluids in dogs is very similar to humans, so that's a great way to monitor it. And those biomarkers are not as naturally complex as in rodents as they are in dogs. And the way that we manage our patients in terms of the medicine and surgical approaches and their therapeutic responses in dogs is much more similar than humans. So if you add this all up, you have a better animal model, and you don't have to use any purpose-bred animal for that either. These animals exist in large numbers, and in some disease processes, it's hard to actually recruit enough cases into a clinical trial. But if you organize and you're interested in finding clinical cases in pets, you can usually find them. Next slide. So the question is, are we really